So let's start. Okay, let's go. Okay, today we are going to talk about the current status of COVID-19. So I just tried to collect some information of current status. Technically speaking, in this question, answer of more than million is also correct answer technically. But if you have more exact and then more useful information, as you can guess, more than 7 million versus more than 1 million is quite different information. Right? So surprisingly, some of you, maybe half of you, thought it's much lower the number right now. In the total world right now, it's more than 7 million by this morning. So a lot of people are infected by this virus. Out of 7 million, more than 2 millions are in America. You can say this is due to the stupid leader. Anyway, it's a very high number in America, 2 million, best country in the world and richest country in the world. But they got this terrible situation. Oh no. Yeah, I made a mistake here, the option of toxicity. At least you could guess, minimum more than 100,000. So far, already dead people is higher than 400,000 all over the world. It's really serious situation. Still, the problem is not fixed yet. Yeah, the best number that we got was about 700, about 2 weeks ago. Unfortunately, we are getting back higher. So this morning, I realized the number is slightly above 1,000. Still, our problem is not fully fixed either. This morning, the number in Gyeongbuk was 19. So Daegu and Gyeongbuk was the worst in the beginning. Gyeongbuk is also less than 100 right now. So that means several hundred in Seoul and Gyeonggi area, maybe half of them, maybe more than half of them is in that area. So the whole statistics is shifting around, but seems like it's it's a quite dynamic situation. Some area is getting better, some area is getting worse. Um, yeah, for now we are okay, but at the end of this week we are getting students back to the campus, so we don't know what is going to happen. Oh, by the way, I checked about the final exam. Um, yeah, you don't need to come to the campus. We can decide. We can decide. So let's see. Maybe too easy. If somebody chose SARS. SARS, MERS, COVID-19, all of them are coronavirus. Influenza is an influenza virus. Yeah, human side, that's a ACE2, angiotensin converting enzyme 2, and virus side, it could be spike protein. And protein, H protein, they are from influenza virus. Yeah, seems like the virus is unstable when they touch the metal, the particle, especially maybe small size of metals. So there are some candidates of this activity, like silver and zinc oxide is also an option. But currently, seems like the old product we see in the daily life is a copper powder. Copper powder coated. The tapes are here and there, as you can see. So gold, very inert. Platinum, not known yet. Aluminum, not known yet. Zinc oxide and silver and copper, they are currently under investigation or in use. Okay, current the standard method is utilizing RT-PCR, not just the PCR, it's RT-PCR. What is RT?
Reverse
Reverse
transcript.
transcript.
Is that reverse transcription or that's a real time?
Both of them.
But here the key is reverse transcription is the key part. Yeah, so far the common vaccines they are composed of proteins. But sometimes we can use the DNA or RNA too. Carbohydrate is not common. When you use the DNA and RNA, what's the principle? How do you use it? Coronavirus is an RNA virus. So in the case that the RNA means this RNA is a coronavirus RNA? You think? If it's a DNA, it can be also the vaccine, then how can you use it, the DNA? Hey, you tell me. Anybody? Someone, you tell me. How can you use the DNA as a vaccine? Wow, I thought I taught everything to you. Okay, there's some holes here, so this can be a bit may consider to study further. So anyway, today we are going to discuss about the current status of the coronavirus and also possible method for handling the problem. They can cover like screening, surveillance, protection, blocking, curing, vaccine, informatics, or anything. So to remind you some of those effects, I just tried to show some of the factors of this coronavirus today. Before you guys start the discussion, okay, I invited the two of you. So Dongwoo will talk about some of those the previous prepared material for DNA sequencing utilizing Nanopore. So Dongwoo goes first, and then Juan will talk about the result of the study of Professor Kim Pineri. Okay. After you hear those other talks, and then refresh your mind with some of those current coronavirus problem, then you are going to start those discussion, what topic we should study further, and which topic you are going to choose for the next week. Okay. So Juan, can I invite, sorry, Tomo first. Tomo, would you start first? Let me
Okay.
start. Okay. Now you can share.
I think you disabled the screen sharing of your attendees, so I think you should allow that.
Now just I allowed.
Okay. Can you see this? Okay, and I'll
Okay.
start my presentation. So I'm going to talk about nanopore technology, and this is the presentation that I made when I was preparing for DNA sequencing week 6. So you have to refresh one for you to comprehend this presentation. So the concept I talk about is about these are the mechanism, the nanopores, and the different features of it, and how we can applicate, and also how it is used nowadays, because it's a method that has been developed a few years ago. So, first,
Your
to
voice
start,
is a little trembling. Could you try to put your mic a little closer and then try to speak more clearly? Oops, where is the one? Okay. Welcome back, Dong.
sorry, my internet connection is not stable right now. I'm in
Yeah,
a
your voice was
rural.
also a little trembling, so you can start again and try to put your mic a little close to your mouth and try to speak clearly.
Okay.
Okay.
Sorry for myself out. So I'll start again, starting from this DNA sequencing. So we all know that DNA sequencing was first introduced by Frederick Sanger, and oops. And this method uses DDNTPs, which are tagged with fluorescent molecules, nucleotides. So after they are put in an electrophoresis, they are separated within different sequence lengths, so we can figure out the whole sequence with it. So it has it has an mm, like a novelty because it was first developed, but it also has disadvantages. It costs very high, and also it consumes very much time. So this is some information on gene termination in Sanger sequencing. So you see that cost is really expensive, almost $2 million, and um, it's used for many applications, but still, this also it utilizes PCR when it sequence the DNA. So we also have to consider more time into it. So sequencing has been developed to solve the problem of the previous sequencing methods. So nanopore technology is a
Hey, 동호.
내 걸로 연결해.
죄송합니다. 제가 지금 그 인터넷 연결이 안 좋은 것 같아요. 왜 이러지?
Your internet problem? Everybody's okay?
Yes, it's my problem. I'm using my own hotspot, so
내 걸로 해. 내 걸로.
너 걸로 써?
In general, it was okay. We could hear you.
I'll connect to another. Okay, so I think it's been solved now.
Okay.
Okay, so sorry again. I'll start with introducing what nanopore technology is. So nanopore technology doesn't require the chemical labeling of the gene, which was introduced in the ddNTP tagging in Sanger sequencing, and also the PCR of sample DNA. And because all of the previous technologies use one of these two techniques, it has been a major breakthrough in DNA sequencing in general. So you can see that there is a kind of a pore protein in here, and the double helix gets unwinded when it comes to this protein, and it gets unzipped. And when it gets through, a certain sequence in here, it is also called as an adopter molecule, and this sort of catches what the DNA strand is, and gives different electric signals depending on what the nucleotide is. So by measuring this different current alterations, we can know that what the exact sequence is. So the mechanism is like this, and over here we could see that it is also using electrophoresis system. So the DNA comes over here, and when it crosses this nanopore, we can know what exactly the DNA sequence on the left is. So this is the mechanism of all NGS. So you can see that there are many different amplicons, which are the same copies of the same strand, and they're kind of cut together, and they're tagged with different primers on their each end, and they get ligated by adapters, and so which has different manuscripts. So it's like different combinations of its break parts. So by analyzing all of them, we can match them together and know the whole sequence. So there are two types of nanopore sequencing, and the first is biological pores, which uses nanopore by using our own organism-derived proteins. So this case in here, it's called MSPA, also known as a microbacterial pore. So this is one type, and the next type is called solid pores. So this is a pore made by humans, which uses very different metal alloys, but it still can do the same. So there are very advantages of both, and scientists are using various ways. And this is called a MinION, and this is a very famous example which uses the nanopore technology to sequence very long sequences in a portable and a real-time manner. So it has been developed by the Oxford Nanopore Technologies, ONT, and you can see that its length is only like 10 centimeters or like that, so it's really small. But we could put chips in there, which are consisted of different DNAs, and by putting on them, the computer just analyzes them and tells us the result. So it uses an CSG, an outer membrane of the E. coli as the porin. So it's a biopole technology. And this is the same technique I've explained before, so I'm going to just skip this one. And this is also the different ways the MinION is used. So first, okay. Yeah, so there's this computer, and the scientist puts like sort of what samples they need, and it gets sampled, and we can create a library which are made by this different kinds of sample ingredients, and we can sequence them, and by measuring these different results, we can know the whole sequence. So the advantages of nanopore is that they're really cheap, and also because they don't use PCR, this does not mistake repetitive sequencing. Repetitive sequences which are located in very many introns, they're really much same to another, right? So they don't mistake them, so it has a comparative advantage compared to other NGS technologies. So the de novo synthesis is possible, which is a synthesis without a reference genome. But it also has a disadvantage that it's not perfect. And one problem is that the background noise of this nanopore technology is very high. So we have to reduce this noise to get pure results. So the National Forensic Service of our country uses this MinION to record many criminals on tracing their genomes. So this is very widely used right now. And also on the right, I have brought some research techniques that are used to kind of strengthen the possibilities of nanopore sequencing. So it says in here that through nanopore sequencing, we could assemble the human genome with ultra long reads, which was originally not possible in NGS. So this is the end of my presentation.
Okay, thank you, Dongwoo. Questions to Dongwoo?
I have a question.
Yep, Yuan.
How can we distinguish the electronic signal by several nuclear exit?
Okay, so on here. So you can see that in G, there's this kind of signal over here, and all the other G's have the same signal on here like that. And the C's are same, and the T's are same. So the computer analyzes this data to automatically transfer these signals into a G and a T. So that's the way it gets converted.
Well, Dongwoo, is that absolute value of a y-axis value, or that's a change of each?
I don't exactly know about that. I
It
think
it
it's
is
a
a
change.
change. If you look at those absolute values, then you can draw more than 4 different the things. So if the string is a move along, then when the new nucleotide coming in, then there is a change. So the change over from previous one to the next one, so that will generate the signals. So you read not the absolute value, how much was the change? You don't see my cursor. So anyway, those increase or the decrease of that, the height gives you the information of ATCG.
And
Dong, you
also,
mentioned that you don't need to do PCR, but still do you need to manage the samples? You add something here. Is it necessary?
Yes, I heard this is the general technique of nanopore. So in here it shows that there are PCR needed, but I think in some cases they don't. So I think like that's a kind of like manipulating kind of changes. So
So you're saying that maybe this kind of a sample treatment may not be necessary, just the DNA itself can be used?
Yes, I think so, because
Okay,
like the whole
other
DNA
questions?
yes.
Other question to Dong?
Yeah, so one thing I'm confused is how this technology is better than NGS. I didn't catch that.
Okay, so because this nanopore technology utilizes the changes of the electric signal, while as other NGS uses kind of techniques that like binds to the sequence itself to retain its information and to add up together. So other NGS, if there is like a single strand that is repeated in all of the introns, they could mistake that for one single sequence in that chromosome. But I think in nanopore, because it like continuously checks all the DNA nucleotides in the whole chromosome, it doesn't mistake one repetitive sequence. So I think that's the difference.
For other NGS, anyway, you need to amplify those clones. So that amplification, if it can be done in parallel, millions of them at the same time, we call them NGS, next generation. But in this technique, in principle, even though you don't do any amplification, just the sample itself can flow, and then it can be sequenced right away as single molecular level. So that's kind of the trick and also the advantage of this method. Now, the DNA has the direction, 3' to 5' kind of directions. So this method can discriminate those direction.
Okay, so I think it can be distinguished because it has to unwind the double helix, and when it unwinds the 5', I guess it can notice that which end they're altering and opening.
Yes, so this protein can recognize those difference of a different the end. Then it can take up the one end and then read to the other end. So this kind of idea has been explored over the last 10, 20 years. So many people try to make this kind of a nanopore, which can allow the flow of a single DNA strand. Now, as you can see, if it is a chemically constructed nanopore, now the direction is one issue. The other one is the opening up these two strands into the single strand. That's another issue. So there were quite the many difficult the challenges and also technical problems. So many ideas, many players have tried this out. Now it seems like this is the surviving one at the moment. And this technique has been used by Professor Kim Pindeni for the next talk of transcriptome analysis of coronavirus. Okay, thank you, Dong. So let's stop here and then let's move to the next talk. And Juan will give the next talk.
Can I share my screen?
Yeah, go ahead.
It is does it the presentation sharing or the
Yeah, I can see them. It's very good.
thank you. Then let me start my presentation. I will present about how can we analyze the transcriptome, which is the setup or RNA transcript of COVID-19. So in May, there was a big issue that Professor Kim solved the architecture of COVID-19. So you can see the first page of her paper at the right side of the slide, and I highlight the and I highlight the red box in 2 words. This is the key method of her research team has been used in this paper. It is DNA nanoball sequencing and nanoball direct RNA sequencing. So I will present about those 2 methods, and let me show you how she compare those 2 mechanisms results, and uh, can be make several discussion about it. So I mentioned about 2 methods, and there is one more method, the analysis of junction spanning rate, which can let us to find the site of recombination. So it is very essential to analyze those transcription. So the first method I will tell to you is DNA nanoball sequencing. As you in the left side, you can see DNB, DNA nanoball. This is the material that the replicated DNA. DNA is messed up in kind of sphere. So at first, you can catch the sample from the infected cell to ssDNA sucker, which have such viral DNA or RNA or such on. So you can replicate it by those such method I called RCR or rolling circle replication. You may heard those method in your life science test in the first grade. So when it is passed through those those process, it may have such tails like it is expressed by 1x, 2x, 3x, or nx in the picture, and those kind of similar patterns of circular DNA is collected in kind of sphere. So those is called by DNB. So this is the kind of several process that how can we make the DNB. First, you need to make a fragment of the DNA sample, and you may select the size of the fragment that you want to make a DNB, so you can be analyze
Analyze.
analyze those sample, and you will attach such tail and adapter to make those fragment into the sucker pattern. So each side of adapter attached to those and into one. So it can the linear patterns of DNA sample can be circular one. So once the single strand circular DNA is is made it, we can use rolling circular replication. So it can be DMB, and we will load the DMB into the plate that the Dongwoo mentioned in his presentation called flow cell. So we can measure such fluorescence in the flow cell. And the second method is nanopore based DRS. This is the method the Dongwoo presented in his presentation. One point that I want to mention in this method is this method can differentiate not only the sequence but also the modification of the RNA in happens in vitro. So this is the picture in the paper written by Kim Neri. So as you can see in the middle of those graph, there are some difference between the black line and the green line. The black line IVT is in vitro transcript control. Otherwise, green line is viral as transcript. So there is such difference between in vitro condition and in vivo condition. So it is referred as there is such modification process right after those DNA RNA sequence is made. So there are a lot of those mismatch in the COVID-19 sequence. So we can estimate that there is such modification in COVID-19. And the last process is the analysis of junction-spanning rate. This is the method that we can check where is the exon site and the intron site. So at first you can make the concentration of RNA samples very high and match those sequences of fragments by such computer algorithm, as you know. So there might be such some mismatches shown in red color in here, because there should be some RNA modification I mentioned before, and those site was eliminated. So we can check out that if there is some other some site that is eliminated or we cannot check those sequences in mRNA, there might be some intron site here. So Professor Neri was checked those exon and intron site in mRNA by those kind of method. So in this graph, there is a computer process wizard that mentioning about the comparison between in vivo and in vitro, and the right side shows the how the recombination of genes is happen in COVID-19. So this kind of this is not a total description of the method that Professor Kim used in her paper, but by those kind of method, she found that how the RNA of COVID-19 is consist of. So this is the end of my presentation. Thank you.
Okay, thank you, Juan. It's a little complicated, but questions? Juan said that this analysis is a transcriptome. What is a transcriptome?
Transcriptome is
No, not you, not you. Doyon?
It is kind of whole set of RNA transcription, including coding mRNA and non-coding RNA.
Actually, transcriptome means it doesn't matter whether it codes or not. If they can be made into RNA from DNA, then it can be regardless of the coding. So all the RNA structure can be generated. It's a transcriptome. So total gene, the gene is one unit of one information. All the gene together, it's a genome. Transcript is a one RNA made from one gene, but if you collect all the RNA, which can be generated by one organism, and those RNA collections, it is called as a transcriptome. And here, those sequencing technique, Nanopore method can sequence not only DNAs, they can sequence RNA too. That's another big advantage. And also he mentioned, Juan, would you go to the epi transcriptome analysis? So we learned about the epigenetics, the previous one. Epigenetics is modified the DNA. And DNA modification, the most famous one is the cytosine, methylated cytosine. Juan, one page back. So here, so depending on those conditions, so there is the red box. That A is a modified A. So we have learned only the methylated C as a representative of most common the epigenetic modification, but there are a lot of changes on RNA too. And RNA is usually unstable. That's why analyzing those changes of structure is more difficult job. So that is also could be detected by this method. So even though we know those whole genome, there are different sizes of RNA can be generated, so-called alternative splicing. Depending on those cleavage site, it can generate many different RNAs. Figuring out exactly what RNA could be made by one virus is very important information. And you can figure out what protein can be made. Of course, if it's the coding of the RNA. So previous case, the quiz problem, we said DNA or RNA as a vaccine. Okay, those DNA, those RNA, they are gene information. They can code the protein which can act as an antigen. Okay, that's how it can be used as a vaccine. You give DNA, not protein directly, but that protein codes the synthesis of a vaccine. Okay, so then the advantage, there may be some advantages. So there can be some topic that you can study too. Any other question to Juan?
I have a question.
Go ahead.
So, so if you look up information in NCBI, we can notice that there are like very many kinds of DNA sequence results from different laboratories. So is this research paper the first one to do that, or is it some kind of different stuff?
It is kind of first. Does my voices come out to yours?
Yes, we can hear.
This paper is the first one that can be analyzed the information about the RNA. So it could be the answer.
Okay, then let's just stop here. Then let's move to discussion part. So from now on, I will shut up here. You guys just start to discuss what kind of a topic would be important topic you may want to study or learn. So try to make out those topic first. And then you decide who takes which topic. Then you are going to prepare for those the presentation material, and then present next week, and we discuss about it. So I recommend the Sejin, you open up one PPT file, and then during those discussion, maybe you can write down, then everybody can see which topic is on the board. Then you can add abstract, and you can decide.
Actually, I prepared this kind
You
of
did already,
file.
okay? That's a nice way too. Go ahead.
Right, so let me lead this just kind of quick discussion. Do you hear me?
Yes.
Yes.
There is some noise, sorry about that. And as it is the last presentation, actually, I want you to prepare as detailed as you can. So first of all, let's talk about all possible topics about coronavirus, and then just gather it up first. So for me, when I read the newspaper, I had a big interest in the research of Professor Junction-spanning in PostSec. So he developed a way of diagnosis using the aptamer. It might be not familiar with you guys. It is very useful tool to diagnosis of coronavirus. So I want to focus on this topic. So no matter who say any topic you have interest, I'm gonna list it up. Nobody say, then this is the order of the age. So Young, you'd be better to say first. As the next senior here.
Can I suggest some things?
Is Dong?
Yes.
Okay.
Yeah, so I have few interests, but I don't know it will be a fun thing to research about. So I brought some topics that could be possible in treating or can be used to diagnose. So the first thing is that I was interested that some patients, some child patients of COVID-19 has this multi-system inflammatory syndrome called the Kawasaki like illness. Did you heard about that?
No, never heard about it.
Okay, so this was first identified in London when kids of COVID-19 were infected. So they didn't show like exact symptom of COVID-19, but like other rather swelling, palms on feet or hand, and other kind of inflammatory disease that eventually caused death. So this could be related to COVID-19. I thought it could be a dangerous problem if our children in our country could be infected and so on. So
what is the name of the disease?
it's called Kawasaki disease.
Saki. Is it correct?
Yes, yes.
It sounds fun. And anybody else? At least you should talk about one topic for each of you. So don't say that I recommended this. And anyone else? I think it might be a simple topic like the current antiviral drug research or corona vaccine, just the professor mentioned, or analysis of virus structure or virus pathogenesis. So it can be anything. If you don't have, I'm gonna list it up first.
Can I suggest one more?
Of course.
In at first, in April, there was the first wave was closely finished in South Korea, but in these days there are a lot of articles that are saying the second wave of COVID-19 is started in South Korea because of clubs or
Yeah,
hospital. Yes, so I want to discuss about how big, how the social effects of social factors that caused the second wave in South Korea. But I think those kind of topics is not that much related to this class. So
No, I think
It's
that's
kind
also
of
fine.
social issue, but it sounds
Yeah, social issue
interesting.
is also fine.
All right, thank you, Joan.
Can I suggest one?
Yep,
Uh,
go ahead.
I'm interested in DNA and RNA vaccine that professor already talked about. It can be many bio company has tried to of this kind of vaccine. So we should learn about that and how can we yes. So I think this is kind of can be a good topic.
Okay, so as I know, the company Moderna is leading to this RNA vaccine as well. So you can research more detail related with this company's work.
Yeah, that's a good idea too.
What about those kind of reusable mask or some pretty fancy technology against the COVID infection? So I suggest reusable, or I can call it the washable mask, was developed recently by Kai's lab. So it can be another good option.
Can I say something? One more.
I think the guys who have never mentioned anything, I recommend first, and then you have a chance to.
Can I suggest
Of course.
for more specific mechanism of COVID-19 regarding to such as NSP, non-structural protein,
NSP?
or NSP, or ORF1A, something like that, and also some idea about COVID-19, such as CRISPR
CRISPR.
genetic seizure or protein seizure.
Yeah. It's kind of gene seizure. I think it's kind of hot topic these days. There are some ideas the virus can kill the coronavirus with use of CRISPR technology. So it might be hard, but interesting. So I don't think Doyun and Sangin never mentioned yet.
I suggest one.
Right.
I first think about Moderna, but if Moderna related with fake spike protein, I would like choose fake something like recombination ACE2.
The combination what?
ACE2.
Recombination ACE2.
Is
So you said you mean it can be another target of this kind of vaccine?
yes.
All right. I have no any idea about that topic, but it might be fun. All right, so Sangin, you are only guy who never mentioned any topic.
I wonder how we deal with mutation of COVID-19. Until now, there is no mutation of this, but this week, one high school student died since she started symptom. Oh, sorry.
Okay. Just keep slow.
It is determined that she is not a COVID-19 patient, but she have destruction of peh and
Lung.
wrong, and she dies so fast. So I wonder, is this a mutation of coronavirus? And I wonder about that.
So you have an interest in that specific case.
And if any other, if there's any other case, I want to study about that.
Okay. So all of you mentioned at least one topic each. So you can pick up the topic you mentioned before, or you can choose another topic suggested by other people. So I think from Dongun to me, pick up topic following the sequence, and if it is overlapped, you can claim why you must pick this topic up. So Dong first. You can pick up topic among this or another interesting one.
Can I pick two, and if nobody else picks one of those two, can I choose one by myself?
Okay. Then
so I want to do either. Okay.
Right. So keep going.
I want to do my own stuff called Kawasaki disease, and also the reusable mask suggested by you.
Kawasaki. you okay to study 2 topics?
No. I mean that if nobody else chooses one of these two, then
one of
I have the
these.
chance to think to choose what I'll do eventually.
Okay.
Hey guys, how about you choose one topic each, then leftover you can manage it later. Also, as a potential topic, this is a kind of a chemical approach of the treatment. So currently there is one chemicals, the drug remdesivir is a kind of actively the ongoing. So maybe comparison of a current small molecule drugs can be important topic. So I suggest the one topic, current status of the small molecule drugs as a potential. Then you can compare who developed how much and then which one is more promising than the other kind of things. So we have heard some of those Japanese one and Chinese one and the Gilead one and several things are ongoing right now. So I suggest one of that small molecule the drug development. Do you have any vaccine development here? Yeah, we have DNA RNA vaccine, but other drug too, other vaccine. Do you have in the list too? So the vaccine is also has a different approaches. Maybe if you didn't cover maybe protein the vaccine, that can be another the possible the topic you can consider. So among those topics, maybe you can choose. Maybe the Sejin said that this is the order of seniority. Maybe you can choose from bottom to up and try to pick up one each. Don't pick up one, then maybe seven.
So Dong, first of all, could you pick one of them first, which is more interesting? Kawasaki disease.
Okay.
Yeah.
Okay, then I'll go with.
Whatever.
Kawasaki disease.
Okay, then Sami.
If Taekjun is okay, I want to presentation about DNA vaccine. Or if he choose this, I'm okay that mutation of coronavirus too.
Okay, anyway, you first come, you can first serve. If you want, you can snatch, but I want you to why you must select this topic. Or you can pick another topic.
Actually, I interested in 2 topics. One
Right.
is DNA and RNA vaccine, and the other one is antiviral drugs, small molecules, the professor mentioned. So I just yes, I want that small molecules drug.
Nice. Then doing?
If it's okay, I want to choose reusable mask.
Yep.
Which
Nobody
is
picked it up.
fantastic. Yeah.
So I think it can be a reusable mask or any other fancy technology against the coronavirus infection. So yeah, it is up to you. Any
Okay.
related topic with reusable mask? Okay, John.
If I could, I want to choose the mutation of coronavirus.
So I think we need to develop this topic more because mutation of coronavirus sounds like very comprehensive. So we need to improve these topics more detailedly. how can you develop this mutation of coronavirus? Need professor's opinion, because in my opinion, it sounds like too ambiguous.
Well, one possible interpretation can be, the impact of a mutation of a coronavirus. Someone briefly mentioned that there is no mutation discovered yet, but already there are A type, B type, C type. Seems like there is some changes are already being observed. So it can give us the information that maybe, the transition, origin, and, which directions it is moving to, kind of thing is also possible. So whether this really the Chinese people is not responsible for that or not. So this can be, some kind of a genetic the tracking kind of the approach is one possibility, or maybe the prospect may be a little too early. We don't have a drug, so we don't have a resistance so far. So it's a little open questions, but that is one direction of that mutation study. So if someone, the Juan would like to take that part, then I think that's one possibility. Yep, that is suggested by Sejin, but it can be a little more explored the broader way too.
Okay.
Juan, is that similar to what you are thinking?
Yes, Professor.
Okay, good.
Right. Thank you, Professor. And next is Young.
Protein vaccine and some important proteins, something like NSP, non-structural proteins that COVID-19 make.
Is this your topic, all right?
Yeah. Protein vaccine is the professor mentioned. I think that it is good topic, and also it can be connected to NSP that I suggested. Yeah.
All right, so actually I have no idea about this topic, so just make a good presentation related with this topic. Okay, and the last one, I'm gonna study Professor John's great achievement about the aptamer diagnosis method. So Professor,
for your reference, the Sejin. So in the campus, actually, there are at least 3 people who are working on the aptamer approach. One is the Professor Zhang, and the other is Professor Song Ryu.
Yeah.
And also the Chang-il Ban in the chemistry. So at least I know 3 people are working on aptamer. I assume all of them are trying to make this the aptamer approach for COVID virus. If you could, then if you can get information, then comparison among their approach is also interesting. And who will be the winner can be another fun kind of things we can talk about.
Okay.
Okay, nice.
This is the end of the discussion.
Thank you, Sejin. So this is the first draft. So please send me this list to me, or you can just share this in the cutoff. So any of you, when you study those material, if you feel it's possible that you cannot collect enough information about them, and if you may want to change your topic. So it's up to you. You can change your topic, but try not to invade others' topic. Okay, you can choose totally new topic, but any changes you suggest that into Katopan. So everybody knows what is the changes, and then the revised the list we can look at together. Then we can decide those order. So who goes first, kind of things. So let's use this information until the Sunday. So let me share my file here. So today we talked about this, and then we got 2 presentations. And next class, we will meet again at the same time, Tuesday at 2 p.m. And the deadline for PPT may be Tuesday morning. But as I said, we are going to continuously modify those topics until this deadline. So if possible, we want to fix those the topic by Sunday. Okay, then you have enough time. Then early Monday morning, I will send you those order of those presentation, maybe 4 talks first, and then 3 talks in the second day. So we are going to try that way. To do that, it's better to finalize the topic by Sunday. Okay, by Sunday it's open competition, the open the discussion. If you want to change, then you are allowed to change your topic. Okay, and the final exam, as I figured out, it seems like it can be done by online. So would you prefer to do the final exam as online, or you want to come to the campus and then have exam together and then go out for lunch together? Some of you mentioned that.
Sounds like...
Sorry.
No.
Some of you, I remember you said there is some plan that you prefer to be the online. Okay, if you don't have the specific preference, then maybe we can do this by online, similar format as we did the last time. So the date and time will be final exam week of Tuesday. So first class time of our class. Seems like on that Thursday was some other holiday. Anyway, so June 23, 2 p.m. will be our final exam. The format will be quite similar to the midterm exam, and the coverage may be mainly focusing on the later part, since we studied those the midterm exam. But I will look back those midterm exam. If there are many errors for some concept, then I will pick up some problem from previous version too. Let's say 70-80% of the new topic and 20-30% of old topics, but something I stressed over and over. Okay. Any question for the presentation and the final exam?
So is the presentation sequence random or follow
That's
any...
something we decide or we decide by Sunday night, depending on those topics. Okay. If it is finalized, then it may be easier to choose. And still, you check those the iClass. So your final the PPT file will be also uploaded to the iClass. So look at it carefully. And if there's any other new announcement, then we can use the Kakao. Okay, guys. Okay. Thank you. See you next week.
Thank you, Professor.